this session. And this talk will be given by Camila Pinto, who's originally from Brazil, and she is now at Northwestern University at the Feinberg Medical School. And she's going to talk about neural and genetic basis for human ability traits. The stage is all yours.
Thank you, Gunnar. 1 second. Can you all see my screen? Yes, okay. So as Gunnar just said, my name is Camila Pinto. I'm a postdoctoral fellow at Northwestern University at Dr. Vania Pikarian Lab, and I'm here to talk about our study on neuro and genetic basis for human ability traits. So we all know, and we saw in the previous 2 talks, that we judge people based on their abilities. We use tasks to evaluate behavior, and we know that these behaviors can be correlated with different clinical outcomes or even prediction to some diseases. But so far, we don't have a common framework to evaluate ability traits. We usually use individual tasks, as Sophie mentioned, the flunker test to evaluate some cognition and attention, and specific other tests for motor or sensory. Here we choose a framework that is the NIH Toolbox that assess these 4 different domains and draw ability traits based on the NIH Toolbox. Why we chose the NIH Toolbox? This is tool that was developed by the NIH and has been using for more than 10 years now. It's used both in the research settings and in the clinical settings. It's very easy to be performed, and it comprises about 38 different individual tasks to evaluate these 4 domains: motor, cognitive, sensory, and emotional. So in this study, what we did, we used 2 big samples. One of them is the NIH normative dataset, which has 1,369 subjects, health subjects with different ages, and the Human Connectome Project dataset, which has 778 subjects. As you see, these are big data sets, and we use those to draw 4 ability traits using these individual tasks. Right. First, we did that on the NIH normative data set, and we used the Human Connectome Project data set to validate these ability traits. Also, in the HCP data set, we have data on brain imaging, and we use that to draw correlates between brain networks and the specific ability traits. And lastly, also the HCP data provides us with family structure in which we have data from monozygotic, dizygotic, full siblings, and no related patients. And with that, we could calculate heritability based of our ability traits. So as I mentioned before, first we draw the ability traits. We use a common data reduction analysis here. We use factor analysis. We also evaluate the number of optimal factors that could explain the variance of these traits. And we end up with 4 traits: motor endurance, emotional processing, executive function, and cognitive ability, and social interaction. Here you can see the individual loadings of each one of these tasks, but for now I think we should focus on the 4 ability traits. As I mentioned, we did this on the NIH normative data set, and we applied the same model in the HCP data set, and then we compare individuals in these 2 samples to see if our ability traits were generalizable. To compare, we built clusters using only the ability traits and not using any other characteristics of these individuals. When we compare the proportion of patients between the 2 samples in the clusters, we can see here in the pie plots that they are fairly the same proportion, especially when we go for age matched in the samples. Why? The Toolbox normative data sample has much older individuals than the HCP, and does help us to see that these patients end up clustering at the same rate in each one of these types based only on the ability traits. We also compare their demographic and socioeconomic characteristics, and they were fairly similar. This means like they were at the same age and fairly similar gender distribution between the clusters when we use the ability traits to separate these patients in different groups, including also level of education and income. Okay, after that, we ask ourselves to what degree can brain properties predict disability outcomes. So we again focus on the Human Connectome Project data. The Human Connectome Project data give us a brain properties in forms of correlation matrix between large brain networks, ICAs, and we regress these brain properties against our 4 latent ability traits. Only 2 of them show significant results in the out of the sample regression, and I'm going to show in the next slide. So one of them was the motor endurance connectivity. We show here that 4 pairs of large brain networks connectivity could predict the performance in the motor endurance ability traits. In blue, we have the model coefficients, and this means that higher connectivity between the default mode network and the visual spatial network correlates with indicates a higher value of a specific patient in this ability trait. And the opposite for the sensory motor and language. Just like to point out that sensory motor here is more related to coordination and speech production. And we show here that the negative relationship between these 2 would correlate with this specific ability trait. Interestingly, if we go to the literature in research trials that focus on understand motor performance or focus only on motor tasks, these are the same large brain networks that are involved with performance in motor tasks. So we believe that this helps to strengthen the validity of using NIH Toolbox as a framework to evaluate ability traits. We also found significant results for executive and cognition and cognitive function trait. And here, 8 pairs of large brain networks entered in our final model. And the same we can see in blue, the blue bars corresponding to the model coefficients, so showing that higher connectivity between the precuneus and the left executive control network indicates higher values of a specific participant on that trait. And again, the same here, the networks involved in this were also involved in other papers that shows that this executive control and salience network are important and associated with working memory in health subjects and subjects, for instance, with schizophrenia and depression. Finally, our last question, also using the Human Connectome Project data, is what if we could estimate heritability of these traits? As we know, we all are better in specific behavior or specific tests, and this has some contribution from our genetic background. So heritability is a way for us to evaluate a proportion of a variance of a determined trait that can be related to genetic factors. There are several models to evaluate heritability, and here we use a simple model that is the ACE model, in which tells that heritability is just a sum of additive genetic variance, common environment variance, and idiopathic environment variance that it could be, it's hard to measure because it's something that varies a lot from individual to individual. And we use data from the monozygotics and dizygotic twins. This model tells us that both monozygotics and dizygotic twins share 100% of the environmental variance since they were born at the same time and these patients live in the same family. But the only difference here would be in the additive genetic variance, since monozygotic twins share 100% of the genetic factors and the dizygotic twins will share about 50%. So when we correlate the pair of twins between themselves, the correlation values, it's an indication of the sum of the genetic and common environment variance. And with this, we can calculate a broad sense heritability. I put the formulas here just for reference, but we can go back if you have any doubts. And we were able to calculate the heritability only when that regression is significant, right? And this was true just for the two same traits, motor endurance and executive and cognitive function trait. This value here varies from 0 to 100, and we show that for the motor endurance, 30% of the variance of these individuals in that trait is explained by genetic background, and for executive function and cognitive function, about 49%. This is very interesting because if you look to other papers evaluating how much of the variance in executive function or in, usually they use working memory tasks, the number is fairly similar. They see that about 40 to 50%, some papers say 70% of the variance can be explained by genetic factors. So again, it validates the use of the NIH Toolbox to evaluate abilities. And lastly, we also went to the factor space and measure the distance between a pair of twins or a pair of subjects in this space, meaning that when we measure the distance of monozygotic twins, they are more close to each other in the trait space, meaning they perform much similar to each other than when we compare with dizygotic twins, full siblings, or non-related family members or non-family members, showing again that there is a contribution of genetic for the variance in these traits. The same thing in the brain space, when we measure the distance between monozygotic twins and compare with dizygotic and full siblings, we see the same trend, showing also for the connectivity between these large brain networks. It's more similar when you have a closer genetic background. So in summary, we used the NIH Toolbox to find these ability traits, and we could find 4 of them, and they validate in another sample. That was the Human Connectome Project sample. 2 of them, motor endurance and executive and cognitive function, were associated with specific between large brain networks function, and they were significant influenced by genetic factors. So thank you for listening to this presentation, and a special thank you to everyone on my lab.
Well, thank you very much, Camila. Thanks for the interesting presentation. Are there any questions from either the audience or from the panelists? Any questions from Sophie or Arthur? Any questions?
Yeah, I have a question. Like, given that I actually work on a similar but different project, but I wonder, did you also consider like genetic correlation across traits? And like, are you also planning to look at this, or have you already written it up? Or
no, we did not look across traits, but that's an interesting question. We look within traits, yes.
Okay. And it was not like the emotional stuff was not heritable?
No, it was not. Also, the sample, if we compare the sample of the HCP data that we use for calculating the irritability and the sample that is usually used in specific big data genetic papers, the sample is small. We have about, like, I think we have about 120 pairs of monozygotic twins and less from dizygotic twins. So we speculate that this could be also a reason why the correlation was not significant. It was kind of a lack of power. But yeah, in the sample, they did not.
Yeah, it would also be interesting to see the genetic correlation within each domain to see if actually these tasks, how, what extent they do correspond or not. Because yeah, that's also always a bit tricky, but like very cool work. Cool.
Okay, nice. Thank you for your comments. We're gonna definitely look into that. Yes.
All right. If there are no further questions, it's almost 45 now. If not, then I'd like to thank all the speakers, and I would like to encourage the audience, if you have questions, please get in touch with the speakers individually. We have actually provided the Twitter accounts as well as the Google Scholar accounts here in the chat box for each of the speakers. Otherwise, I'd say thank you very much for the nice session, and I wish you all a very nice day. Bye-bye.